There we go. Any comments, concerns, suggestions? I know there's some issues with the syllabus quiz. I'm basically, giving students credit if they write on the questions that don't actually have correct answers. That's my bad for things that I changed up a little bit in the syllabus and forgot to change the syllabus quiz. That is definitely my bad. But just do your best, and everything will all work out in the end. So today we are going to be continuing with Chapter 1. Chapter 1 is kind of a big overview, as we mentioned before, and I don't really want to dwell too much on Chapter 1 because it is just a review chapter. So, an overview chapter, obviously not a review since you haven't had the course yet, but it kind of reviews some things that you've already covered in other classes. And if you were here on Wednesday, we kind of had a nice introduction to some of the more bigger advances in genetics that you probably know about. But talking about cloning, and why is this not working? There we go. And things about DNA is just a molecule and we can manipulate it as we need to. Well, now we're going to kind of get into not really DNA as the molecule, but more of DNA as the unit of heredity and variation, as we mentioned. So let's get back into that area. All right, there we go. So this is where we kind of left off on Wednesday. We didn't get very far. So when we talk about the study of heredity and variation, we needed to define those two terms. And we took a long time to define what heredity means and what variation means. And so these two ideas, these two main concepts are kind of overarching across the whole course. And we're going to be looking at these two aspects. First of all, the heredity aspect in the first half of the course, and then the variation aspect in the second half of the course. Now, we are by no means going to cover everything in genetics. Genetics is huge, and each one of these sub-disciplines would be a semester class. In fact, you could stretch it out to a year-long class if you were really, really good at teaching it and really knew your stuff. So there's a lot to learn about genetics because it is modern, but it is kind of showing us so many things that are possible that we never thought possible before. And so I always think of genetics as kind of an optimistic science because it more opens doors of possibility rather than closing doors of, we already discovered that, there's no need looking at it again. So every time you study genetics, you find special cases and exceptions to the rules, and that's what's really exciting. So each gene kind of works by itself, and I love that about the field. Everything's always new, always something new to learn. All right, so I mentioned that we're going to be reviewing a little bit of what you already know. And just to kind of review from your 120 and your 201, that all cells pretty much share their chemistry, and that's because they're all made of the same stuff, right? Just like we're all made of atoms, that's all the same stuff. We're also all made of energy, and we're all made of organic molecules. And so to be living, you have to be made of organic molecules, and we know that there are 4 basic types that we kind of divide them into based on kind of the roles that those molecules play in the cells and in the organism. Now, a lot of our molecules that we're talking about are very large molecules, macromolecules. And they become macromolecules not because they're made as macromolecules, but because they are synthesized in bits. And those bits are called monomers. And the monomers, no matter which one of these macromolecules you're talking about, these monomers are arranged in a chain-like fashion to create a polymer. And so that mer, that MER little root back there, just means unit. And so it's a repeating unit. So a monomer would be one unit, and a polymer would be multiple units all linked together in a chain-like fashion. Now, in organic molecules, they're linked together using covalent bonds. And as you may remember, covalent bonds provide a source of energy in the form of chemical energy. And our body uses all that energy, blah, 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 to sustain itself and then to make new molecules. Well, with genetics, we're mostly concerned with two basic types of molecules, and that is the nucleic acids, the DNA and the RNA, and also the proteins that are made using the information in the DNA and the RNA. Now, occasionally we talk about carbohydrates or lipids when you come to those special cases where you may have a protein enzyme that works on a carbohydrate or a protein enzyme that works on a lipid that may be missing. And then we kind of get interested in carbs and lipids. But for the most part, we're really just, in genetics, really just talking about the DNA, the RNA, and the protein. And we'll come across some chapters kind of at the second part of the semester where we're talking about how these molecules are made, like the actual processes that make nucleic acids and the actual processes that make protein. And this, you already know what that's called. That is actually the central dogma. And so the central dogma just reflects how that information works in the cell. So we'll look at that later on when we get to those chapters. Now, as you already know, when we're talking about DNA, which is the main molecule of genetics, because that's what genes are made of, the main molecule is made up of individual nucleotides, and these little nucleotides are basically some kind of base, nitrogenous base, that's attached to a sugar and a triphosphate. And that carries the energy, just like ATP carries energy. These nucleotides carry their own energy with them for assembly into the nucleic acid. And so we have these building blocks up at the top, and I know it's upside down, but it's kind of the way I think is kind of upside down sometimes. And I preferred thinking about it from going from the small to the big rather than from the big to the small. And so in this case, we have our nucleotides. Here we have cytosine, if you can read upside down. Here we have guanine. And we know that those are paired across from each other. Here we have them paired across from each other in joining hands or hydrogen bonds in the middle. And we have, if we have enough of them, we wind up with a nucleic acid. And in DNA's case, its natural cellular form is going to be associated with another strand, another polymer of DNA together to make the double helix. And so we'll study the shape and the structure of the double helix, the current model of thinking of what that actually looks like. We can't see nucleotides, we can't see the DNA double helix structure, but we can get a lot of indirect evidence as to what it looks like. Now we have these really long strands of DNA. In fact, they're so long that sometimes you can see them in electron micrographs. They're really, really long and string-like. But if you're talking about a eukaryotic cell, which we often are in this class, because eukaryotic genetics is slightly more interesting than prokaryotic genetics, the DNA is going to be wrapped around a bunch of proteins to make it shorter. Like if you knit, you're not going to leave your yarn just kind of free in a big glob. It's going to be wound up into a ball, so it's easier to kind of pull a little bit out and it doesn't get tangled and broken. So we have it kind of wrapped around these little protein balls so that it can assemble inside the cell. And then in special times of the cell, that assembly of DNA and proteins, which we call chromatin, they are going to be sort of wrapped up even tighter to make mitotic chromosomes. But before we get to mitotic chromosomes, which is what we actually see here, it's a little bit misleading, we have regular chromosomes that are just at this level. And they're kind of looped in there, and we'll talk about what the DNA actually looks like when it's just in a cell. So we have our chromosomes, really long pieces of DNA, and those chromosomes are going to be fitted inside a nucleus, and that nucleus will be inside the eukaryotic cell. Now, if you're a prokaryotic cell, the DNA looks slightly different, and we'll look at what that looks like in later chapters. So don't worry about that yet. Now, when we talk about proteins and the role of proteins in cells, proteins are what do the work, right? Because there's work to be done in a cell, whether that work is a chemical reaction or that work is moving things, transport, or delivery, or whatever kind of work has to be done in a cell, it's usually accomplished by a protein. There are some exceptions, of course, but usually we're talking about proteins. And so the proteins are going to determine what the cell looks like, what the cell does, what the cell's role is in the tissue, in the organ system, in the body. And so these proteins are quite varied depending on their role, because remember, form leads to function. And so this function is what we need. And so the function is selected, and because that function is selected, that was the form that the protein took, which means that's the information that the gene had in it that led to that form. So these proteins, because they're expressed from genes, reflect the information in the gene. And that's why we look at proteins as well. So geneticists are really involved in protein structure and protein function and things like that, because that's the gene product. So we're looking at the genes themselves and the products that they actually produce. Now, in any cell, not all proteins are going to all be made at the same time, because if you think about a body, every single cell has the same set of DNA, which means it has the same set of protein instructions. But not all cells are the same. So that different cells use different proteins at different times. And so part of the wonder of the genome, and what we haven't quite figured out completely yet, is how each cell can kind of monitor their own set of proteins and keep it completely different from another cell's set of proteins. So that regulation aspect is really exciting to a lot of geneticists and genomicists. That's actually a new field now, as to how the genome is actually, all of the genes work together. Now, when you talk about a set of proteins in a cell at any one time, say the genome creates this set of proteins because this cell has a specific role, then we call that the proteome. And that's an interesting word. And actually, that is more informative in terms of disease and health, because it's the proteins that do the work, and if the proteins aren't correct, they aren't correctly folded, or they aren't correctly expressed, or maybe the gene isn't expressed enough to make enough protein, whichever that is, the protein is what leads to the phenotype. And so we care more about the protein nowadays than the gene itself, right? And so we look at the protein, and if the protein is different than it should be, then we start looking back at the gene and see what happened. So it's a different way of thinking. It's thinking about the phenotype first and then going backwards to the genotype. So geneticists have had this whole kind of reckoning about how they do their science. So you'll see a lot about proteomes if you actually look into genetic research. Now, an example of a proteome protein would be over here on the left. We have an example of a tubulin microtubule. So tubulin is the protein. There's an alpha tubulin and a beta tubulin, and they assemble together to make these big, long microtubules. And so it takes two different genes to actually make a microtubule. But a microtubule's function is to allow transport of things in the cell using myosins. And I'm sorry, using kinesins and dyneins. Sorry, actin uses myosins, as we see over here. So here we have our microtubule. We have a little bubble of something that needs to be moved. It's attached to the microtubule, and it can walk along the surface of the microtubule. Now, that's a really important protein in nerve cells, where you have these really, really, really long axons that are actually just a bundle of these microtubules. And so they grow as long as they need to grow until they connect to another cell. And once they connect to another cell, these vesicles kind of move the neurotransmitters all along the axon so they can create that signal at the end. And so you'll see more microtubules, say, in a nerve cell axon than you would in just a regular, any other kind of cell. So basically, nerve cells produce tons of these microtubulin, these alpha and beta tubulin proteins. Now, muscle cell has very specific proteins that you don't find in other types of cells, and that's myosin, specifically muscle myosin. Myo means muscle, so obviously anything that has myo in front of it is a muscle-specific protein. And so here you see kind of part of a sarcomere, just a little tiny sliver of a sarcomere, where you have your myosin filament here, your thick filament, that's going to grab these actin fibers and slide. And that's the sliding across. When they slide together, the actin slides together, that creates contraction of the muscle. So if you need to pick something up, and then as you relax the muscle, the actin slides apart. And so it's the sliding of the actin in across the myosin for contraction, the sliding apart for relaxation. And those proteins are very specific. They are part of the proteome of the muscle cell. Now, other types of cells are singular in that they're the only cell in the body that actually produce a protein. And that's an important aspect of trying to correct deficiencies of proteins. For example, this is a picture of the insulin hormone, which is a very small protein. It's so small that we actually call it a peptide, because it's not really big enough to be a protein to do work. And so, excuse me, the insulin hormone is used for signaling. And it's used for signaling to allow glucose to come out of the bloodstream and into the cells. And so the cells can actually use the glucose or store the glucose, whichever is needed at the time. So the insulin hormone is really important, but it's only produced in a very small population of cells called beta islet cells in the pancreas. And if you say you acquire diabetes type 2, usually that's an autoimmune issue that kind of destroys specifically these beta islet cells. And if you destroy all of the cells, then you have destroyed your ability to produce insulin. And so you become dependent on insulin shots. Well, geneticists said, that's very interesting. There's no protein made. We need to make the protein. Every single cell in the body has the gene to make the protein. It's just not turned on. So if we can figure out how to turn it on in another cell, then we can produce the protein again. And so they've actually figured out how to express insulin in alpha islet cells, which are right next to the beta islet cells. And so if they're able to produce insulin from the cells that are still remaining, the living cells, then the pancreas can produce the insulin again, and you don't have to rely on artificial insulin. It's a really cool way to think about disease and correcting disease. And it's not a really invasive procedure because it targets a very small population of cells. But I think that's a really cool, very specific application of a type of gene therapy. Now, we mentioned doing work, and other proteins do work in specific ways, but enzymes do work in a way that changes chemical structures. And in that way, they require some activation energy, just like a chemical reaction, and they just become kind of the site of the chemical reaction. And so the enzymes have a very specific role in actually literally catalyzing chemical reactions that wouldn't happen, that they're spontaneous, but they're not as likely to happen if the enzyme weren't there. And so the enzymes are there specifically to catalyze reactions. This is a molecular model of an enzyme called catalase. And catalase is an enzyme that is very commonly found in peroxisomes. And peroxisomes are there to basically detoxify hydrogen peroxide. Hydrogen peroxide is a toxic byproduct. It's very corrosive. It's a toxic byproduct of cellular respiration. And so if you do a lot of cellular respiration, say your body is highly metabolically active, say you ran a marathon and you've depleted all of your energy stores because you've been using energy for hours and hours and hours, you're building up a lot of toxic byproducts, not just the lactic acid, but also a lot of peroxide. And so the catalyze or peroxidase is going to take care of that for you and get rid of it so it doesn't actually damage your tissues. So very important enzyme. Now we talk about DNA as an information storage, and then we talked about protein as the end product of what that DNA tells us. But a lot of students don't really get introduced to kind of the structure of the gene and how the gene is actually information, right? We know it's made of DNA, but what is it about the DNA that makes it information? And so through the class, we're going to learn some of that. We're going to learn the basics, but we're not going to learn everything, obviously, because we just don't have enough time to look at gene structure. I mean, I could spend weeks on gene structure, but we are actually a shortened semester this year. We're only 15 weeks instead of 16. So I don't have time to go in depth into gene structure. But this image that I'm showing you right here is a summary, basically, of the pathway between all the way from the gene itself to the folded protein to the actual parts of the protein that are important for the action, the work that's going to be done. And so it's a very amazing summary figure. Now, this is a gene called CF1, and CF1, up here you can see the gene CF1, and if you remember that the names of genes have to do with what the protein does, right? It has to do with the function of the gene product. And so CF1 stands for complement factor 1, and complement factor 1 is expressed in the retina, which is the neural layer in the back of your eye. It's what interprets the light that you see and sends that message directly to the brain. So it's hooked up to the brain. Now, if this complement factor is not expressed correctly, it can lead to a disease condition called macular degeneration. Macular degeneration is a common age-related change in the retina. In fact, if you spend a lot of your life in bright light, your retina is doing a lot of work. It's getting damaged daily, and it needs rest. And if you don't give it time to rest, it can actually get a little bit damaged. Now, the macula is the very center of the retina, and that's where you see very clearly. It's your perfect pinpoint vision. And if your macula gets damaged, then you don't see very well. So this is a form of age-related blindness. It doesn't happen to everyone, but there are some genetic risk factors for it, and one of those risk factors is mutations in this specific gene. Now, if we look at the gene structure, it looks like a line, and that represents the DNA double helix. And so this line schematically has these little kind of hash marks, these vertical ticks, and we have a 1 over here, and then we go a long way, and then we have a 2, and 2 is a little bit thicker than 1, and then we have a 3, and then we go all the way to 13. Well, these numbers represent the exons. Do you remember exons? What's the other part besides exons? Absolutely, the intron. And so this bit right here that has no number on it is the first intron. And you can see that that's a huge intron in comparison to the, and the size of these little ticks kind of tells us the size of those exons. And so the coding region of this gene is actually quite small compared to the overall size of the gene because the introns are so big. And so that's something that's really interesting to geneticists. Why is the intron so big? Now, if you transcribe that gene so that you're just taking the exons, getting rid of all the introns, what you wind up with is a schematic mRNA down here. And so here we have 1, which corresponds to exon 1. We have our 2, which corresponds to exon 2. So we've gotten rid of this big intron. And then we line up the rest of them all the way to 13. And what you may notice about this diagram is that it is color-coded. And the reason it's color-coded is because certain exons correspond to certain active areas of the protein called domains. And so here we have a domain that's orange, and the orange domain comes from exons 9 through 13. So 9 through 13 over here encode the last bit of the protein. And so we also have other functional areas here, LDLR2, LDLR1. And so these are like the LDL receptor. That's what the name means, LDL-like receptor. And LDL receptor 1 and LDL receptor 2, they're like those two receptors. And that's for cholesterol management. Macular degeneration is associated with pretty interesting. We have a CD5 domain. I don't really remember what that means. And this FIMAC domain. So these probably have to do with localization or where the protein is in the cell. And then this one that has the ACE on the end, this is the part that's the enzyme part. And so the enzyme part is what does the work, even though the rest of the protein is just there maybe to stick in the membrane or to receive messages, receive signaling from other molecules. And so there are different areas of proteins that do different things, the domains. So one thing that geneticists are interested in is where do the mutations occur? You know, is it in the enzyme domain? Is it in the binding domain? Is it in the localization domain? All of these things that are important to know about your protein and where your mutation is. So if you have a mutation, for example, in a localization domain, the protein doesn't go where it's supposed to go. If you have a mutation over here in the enzyme domain, depending on where it is, and in this diagram here, we're actually showing which ones of the individual amino acids are most important. If you have a mutation, say over here, it may not matter. But if you have a mutation in the active site, then that's going to basically shut down the protein or shut down the enzyme activity. So we look at all aspects of the gene, all the way from gene structure, all the way through to not just the protein itself, but the active areas of the protein, the important areas of the protein when we're looking for mutations. So it's really sort of a mystery, sort of a you know, like when you watch like a mystery show, like a I like to watch old British murder mysteries, and you don't quite know where they're going, but you get clues along the way. And so that's kind of what we're doing when we study a gene. It's really kind of searching for the mystery in this specific case. We know the general rules. We know what to look for, but we don't really know exactly how this gene is doing what we think it's doing, right? So each individual gene has its own specifics about it that make each discovery really exciting. Now we mentioned nucleotides, and we mentioned that the nucleotides are the information, but it's not just the nucleotides, it's the sequence of nucleotides. So what is it about the nucleotides that provides sort of a language? So the language here comes from those nitrogenous bases, and they're called nitrogenous. That just has to do with nitrogen and we put an adjective on the root on the end. So these bases have a bunch of nitrogen in them. And that's why it's important that you get, nitrogen in your diet and blah, 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 and you get that from proteins. And, if you have excess proteins, they just get excreted in the urine, and, it's the nitrogen. Now, when you look at DNA, the most common nucleotides that you will find are adenine, thymine, cytosine, and guanine. Now, occasionally you can find others. Sometimes you might find uracil in DNA. Sometimes you might find chemically modified forms like methylguanine or methylcytosine, different forms of those. But these are the most common and the ones that we usually see when we're thinking about sequences, especially coding sequences. So the adenine, thymine, cytosine, and guanine act as sort of an alphabet. And so you can make words, but your words can be, 100 letters long, or 1,000 letters long, or 10,000 letters long. And the uniqueness of that comes from the sequence of the individual nucleotides within that stretch of letters. And so if you think about, you could have any 4 nucleotides in each one of those positions, and you're talking about 10,000 letters, you could have unlimited sequences. I mean, there's so many different possibilities. I mean, obviously there's a limit. If you do the math, you can figure it out. But so many possibilities for sequences that we have unlimited potential to make proteins. And that's important to evolution and variation, that we have unlimited potential. But, as we'll see later on, the genetic code kind of takes away some of that ballast, or, some of that power. So the storage of those nucleotides in a linear sequence with covalent bonds in between means that they're kind of mostly permanent and unchanging until a mutation occurs and changes it. But for the most part, it gets passed down as the same information as that molecule is copied and passed down. And we'll look at some chapter with mutations and how mutations can occur and change the sequence as well. Now, we keep talking about DNA as information. One thing that was just kind of groundbreaking in terms of understanding how DNA is information was the cracking of the genetic code. And the genetic code was cracked in the late 50s, early 60s. And what they did is they figured out how the ribosome actually translates messenger RNA. So it's not necessarily what's in DNA, it's what's in the RNA that matters. So if you have a DNA sequence of, for example, these 12 letters, then that's going to make RNA with the same sequence. Copy it to the RNA. And then if you use the genetic code to translate it, then you will get, for every ATG sequence, you get a methionine. So when we think about A's, T's, G's, and C's as letters in the alphabet, we then arrange them into words of the genetic code. And so the words within the genetic code that are basically 3-letter words, those words are the vocabulary. And so the vocabulary is what we can make our sentences with. That's how we can make our proteins. And so it kind of limits us in a way that now we don't have 4 possibilities. Now we have these sort of 64 possibilities of different words. Now, the cool thing is that that's one way to read it. So if you start at nucleotide 1 and you go 1, 2, 3, then that's a word. But if you start at nucleotide 2 and go 1, 2, 3, so if you start at A and go ATG, you get methionine. But if you start at T and you go TGG, that's something completely different. And then if you went GGG, that would be different as well. So each gene actually carries 3 possible interpretations of the information that they're carrying as well. So look at all this possibility of information. It's amazing. There's so much possibility out there for the genetic code to carry information and to give that information to the cells as they need it. So my question down here, where are the codons and what reads them? Does anybody remember what reads codons and makes sure that and actually translates them? You have to kind of think all the way back. tRNA, absolutely. So ribosomes don't actually do anything other than stick. So there's a little ribozyme that sticks the proteins together, that builds the proteins, but it's the tRNA that does the interpretation, absolutely. So the combination of the mRNA, the tRNA, in context of the ribosome is what's actually doing the work of translation. It's a really amazing, beautiful process of everything coming together and all the partners doing their jobs. I like that idea. All right, so with all this information, we have these long pieces of DNA. Now, realistically, long pieces of DNA can get tangled. And so, as I mentioned before, most DNA is going to be packaged in a way that it's going to be further protected, besides just having the two sides of the double helix protecting it, protecting the information inside, the base pairs. It also has these proteins that are going to help create sort of a scaffold for the DNA double helix so that it is further protected and it can be kind of packaged inside the cell. Now, for eukaryotic chromosomes, that's what those long pieces of DNA are called. For eukaryotic chromosomes, we talk about them in terms of chromatin. And so chromatin is a special word that means a eukaryotic chromosome that's wrapped around histones. And once it's wrapped around histones, it can also associate with other types of proteins that help it kind of loop and stick inside the nucleus. And we'll look at a nice diagram of that later on when we start talking about chromosomes. But for the most part, here we see a really cool picture. This is an intact nucleus where all of the chromosomes have been stained different colors. And so you can kind of see here's a green one over here, and here's a red one over here. We've got a kind of an aqua one over here. And you can see they kind of occupy different areas of the nucleus. And then here, if we kind of burst open a nucleus and look at the chromosomes, you can kind of match them up. So here's a green and here's its partner green. We have two oranges that are partners here. We have a couple of reds that are partners. And you can see them and see that the chromosomes themselves have characteristics that make them a little bit different than others. Like we have a really tiny orange one here, which that's either 21 or 22, I can't tell. And then we have, longer ones like this, like this red one over here, or like this big yellow one, which is actually chromosome 1. And so if we arrange them based on size and shape and all that stuff, we can actually pair them into a karyotype. A karyotype is just a picture of chromosomes. And we see that this individual has the right number of chromosomes, 2 of every type. Eukaryotes being diploid, that means that they have 2 sets of each chromosome. And then we have 2 X chromosomes down here. So we can actually also predict that the sex of this individual is going to be female. Now I say predict because we don't know the phenotype of this individual. Chromosomally, we predict that they would be female. Now, an average human chromosome contains tons of nucleotides, which means that any one chromosome could have thousands of genes like embedded within that chromosome, within that sequence. And so any one chromosome that gets passed from parent to child, and if you think about your diploid status, half of these chromosomes came from mom and half of them came from dad. And so you got that chromosome, it was passed to you during fertilization. And so when you get that one chromosome, you get all of the genes that are on that chromosome. So you kind of get these inheritance groups. And so heredity is kind of complicated by the idea that you get groups of genes together on your chromosomes. And we'll look at that. That's a phenomenon called linkage. We'll look at that kind of in depth. All right. So, time to sit back and think. What types of macromolecules would you find in a chromosome? Is it A, polysaccharides, B, proteins, C, lipids, or D, nucleic acids? Absolutely. Good. I'm glad everyone's answering D, because obviously a chromosome is the storage unit of nucleic acids. But also, and someone said it, a couple of you said it, proteins, right? So you can't fit inside a nucleus if you don't have the proteins to wrap the nucleic acid around, right? So you have to be able to package it inside the nucleus and protect that DNA sequence. And so it's very important to have your proteins. So sometimes when I do one of these concept checks, I kind of leave it open for a discussion as to, you know, is there only one answer, are there two answers, etc. So as we go, there's always a possibility of two answers or three answers or no answers. So it becomes a discussion point. Just FYI. All right, so I've only got less than 10 minutes left to work on the rest of the chapter. So we'll do what we can do, basically. So when you talk about genes and their purpose, so the purpose of genes is to contain the information to make gene products. We rely on this aspect of gene expression, right? Because just because you have a gene doesn't mean that you're going to express it. Think about someone who has type 1 diabetes where they never had the ability to express insulin. That means that they may have the information, they just can't express it. And that means that they don't have a phenotype because they have somehow lost the ability to create the phenotype. So gene expression is kind of the link between the two, the link between the information and the phenotype that comes out of the information. Right. So the whole process of gene expression is the central dogma that you have been learning all along, where you have DNA goes to RNA, goes to protein, right? So we'll look really in depth at those two steps and see what can happen during transcription. And that's kind of the last thing that we talk about in the semester because it's the most complex. And so we leave that until we've learned everything else to then start talking about transcription differences. So how we can change expression at the transcription level. And then there are also ways that we can modify expression at the translation level, like preventing translation from happening or misfolding the protein or something so that even though you've expressed it, it still doesn't create the phenotype that you want. And so when we talk about genetics and genetic diseases, it's not necessarily the gene that's the problem. The problem is somewhere between the gene and the eventual product, the phenotype, right? So the phenotype is the eventual outcome of the genetic information. And so for a normal, quote unquote, normal phenotype, in fact, all of us are on a spectrum, but you know, for every phenotype, a quote unquote normal phenotype would require a lot of steps to get there. And so we look at all of those steps in the journey between information and phenotype, or genotype to phenotype information. Well, that's basically what genetics is. It's the journey from genotype to phenotype. All right, so when we look at these two sequences, And we are thinking about the idea that information is stored in the sequence. If you look at these sequences, are they the same? I mean, we can obviously look at this and go, okay, A and A are the same, but no, we diverged in the third nucleotide. So if you look at these two and you think, if these are two genes, are they going to make the same protein or different proteins? Anyone have a guess? You can be right or wrong, doesn't matter. Could be different because the sequences are different, right? So what information do we actually need to see if these are the same or different? What information is missing? What's going to tell us what the protein will be? Absolutely, the codon table, right? So if this is transcribed to RNA and then translated to protein, we need to know what the codons are going to tell us. You know, are these words the same or are these words different? And so just as we know that, you know, two different words can be spelled differently and still have the same meaning, I know that's not completely true, but if you think about kind of popular words, you could spell them differently and everybody knows what you're talking about, right? So you can interpret them differently. So what we actually need is the genetic code to tell us if these proteins are in fact the same or different. And so if we start, I'm going to activate my pencil here. If we start on sequence 1, we have AAG, and we go to our first letter A, our second letter A, go to G. Here we have lysine. So that's the first one. If we go here to our second sequence and we look for AAA, we go A, and A, we have lysine. Kind of interesting. All right, so let's go to the second codon. CGT, we have C, G, and here would be U because we are an RNA. So CGU would be arginine. And here we have CGC, different sequences, but you go CGC, and we have arginine again. And so if we went across all of these, we would see that we have the exact same protein. So if you have two different sequences that make the same protein, how cool is that? It's really cool. So just because you have a mutation doesn't mean that you have changed the protein. So looking at mutations first is not the way to determine whether you're going to have a problem in the phenotype. The way to determine if you have a problem in the phenotype is to look at the phenotype, right? So in genetics, you always start with the phenotype. In fact, as we go through transmission, that's where we will start. We will start with the phenotype, and we will then look at inheritance based on offspring phenotypes. And so we're going to kind of track phenotypes back. When we talk about transmission genetics, we're usually talking about traits and some other terms that are very specific to the experimentation that we'll be learning. Those are going to be your terms that are in Chapter 2. So as you prepare for Chapter 2 next week, for Monday, just remember that we're looking at phenotypes and we're interpreting genotypes based on phenotypes, okay? Now, I know I haven't gotten through this whole chapter, so what I will probably do is just kind of make a video to post that is the rest of the chapter, so that if you have, because I know that there are some homework questions based on the second half of the chapter that I haven't covered today. So what I'll do is I'll make a video of the rest of it, and then you can watch it at your convenience if you need a better explanation of what's going on other than the chapter. Okay, sound good? And I am out of time, and my cat is yelling at me to feed him. So any questions, concerns, comments? I appreciate all of y'all coming to class today and learning with me. And I will see you on Monday. Have a relaxing weekend. Get some good sleep. And be ready for Monday, because we are going to pick up and learn some stuff. All right. Have a great weekend.